Hi, everyone. Sorry about the delay. We were having a little bit of a challenge getting everyone in place, but we are here now. Thank you so much for joining us. As a reminder, we'll have question and answer session later, so please feel free to enter your questions in the Q&A section of the webinar. Friends hosted a cell therapies meeting with the Parker Institute for Cancer Immunotherapy on May 6th. And so today's advocate-focused meeting will provide you with some more information and allow you to ask your pressing questions about cell and gene therapy. Today, we are very lucky to be joined by Dr. Alex Marson from UCSF and patient advocate Chris White, both of whom were panelists during the May 6th event. After they introduce themselves, we'll ask them each to share a quick overview of their work in cell therapies. So first, Alex, can you give us just a little bit of background about yourself?
Sure. My name is Alex Marson. I'm a professor at UCSF, and I have a lab that studies the immune system at the Gladstone Institute here in San Francisco. And we have been interested for a long time in how the immune system works, but over the past 10 years, we've seen this enormous opportunity to think about repurposing the immune system as a drug to treat disease, especially cancer. And so that's become the major focus of our.
Awesome. Thanks, Alex. And Chris, will you do an introduction of yourself?
Absolutely. My name's Chris White. I'm based out in North Dallas. I'm a mucosal melanoma survivor, thriver, advocate, and self-published author / inspirational speaker. And it's a pleasure to be here. I've been fighting mucosal melanoma, diagnosed in 2018, and over the course of 18 months, finally got into a program for TIL therapy, and within less than a year, had a complete metabolic response. And here I am.
Thanks, Chris. And now we'll ask Alex to share a little bit of a presentation about his work.
Sure. Let me see if I quickly share my screen here. So I thought I'd give just a little introduction about my work. And sorry, is that working now? I assume you're seeing my screen. Yeah, so I'll give you a little introduction about my work and the approach that we're taking to build new immunotherapies for the cancers that are still not yet responding to immunotherapy. So I love this slide. It was made by someone here at UCSF. I think it captures the spirit of what we're trying to do. We're really trying to take cells in our immune system and use the new tools that are available to us, especially CRISPR, which I'll introduce, to go inside of these cells and get at their DNA source code, the code that will determine all of their functions, and rewrite it to put precise instructions into these cells to make them into cellular medicines that will be safe and effective against cancer. So we're making progress, and there's still a way to go, but I thought I'd introduce this. I should say that I'm involved with a number of companies that are working in this space and related space, especially Arsenal Bioscience, which is now a clinical stage company in clinical trials for ovarian cancer and kidney cancer, taking related technologies forward for treating patients. Just to give a little bit of a sense of how fast this is going, I thought I would start here. 2012 happened to be the year that I came to UCSF. I finished my internal medicine residency and came here to finish up training. I was actually training in infectious diseases. And two things happened in 2012 that really changed the course of my research and the course of so much of the field and so many opportunities for immunotherapy. One happened in Pennsylvania. Carl June enrolled Emily Whitehead as the first pediatric patient to receive CAR T cells. And I'll say what that is in a second, but basically she was enrolled in a really experimental treatment where her cells were genetically modified to redirect them against her leukemia. And she went from really not having any treatment options to being cured, and she remains cancer-free. So this really woke the world up to the possibility that genetically engineered cells could make a huge difference in cancer. And at the same time, in the same year, Jennifer Doudna and Emmanuel Charpentier published this paper that ended up winning them the Nobel Prize, that has a complicated title here in science, but was the first paper that really showed that CRISPR could be used as a tool to change DNA. And so, I think there was this amazing convergence that opened up. All of a sudden, cells could be medicines, and we had a tool to go in and actually program these cells to do what we want. And so that has been the direction of research in my lab for the past 10 years, opened up by that convergence. Just to briefly introduce what CAR T cells are, this is a schematic of the type of CAR T cells that Emily Whitehead and now many, many others have received. Patients largely go in and will donate a large blood sample that gets sent to basically to a factory that will genetically manipulate them in clean conditions. That's currently done with these viruses that insert DNA material in a relatively haphazard way. But get these instructions to add a new piece of code so that DNA gets inserted into the genome of the T cells. And that gives instructions to make an artificial receptor called a CAR, which stands for a chimeric antigen receptor. Those cells can get infused into a patient, and the results can be really miraculous. And I'm showing here just some of the FDA-approved drugs that are now CAR T cells. These are drugs that are actually cells. They work remarkably well for certain types of leukemia and lymphoma. Still have room to go to find what's keeping some patients still relapse or don't respond. And we still have a huge way to go for the more common types of cancer, solid tumors, breast cancer, colon cancer, pancreatic cancer, brain cancer. So we have a lot to go, but we are very optimistic that we're just at the beginning of what T cell therapies are capable of. And that's because of technologies like CRISPR. With CRISPR, we can really be much more precise and comprehensive in how we think about putting code into DNA to make cells do exactly what we want. We'd like to program them to be able to go where we want, find cancer, clear it, and make sure that they don't have collateral damage of damaging any of the other cells in the body. And so we think about, being able to pick precise places in the genome and make really programmed changes to the DNA. So this CRISPR is the tool that has allowed us to do this. I think I'll breeze through a little bit of this intro, but just wanting us all to be on the same page. The CRISPR is basically a scissor that we can program to cut at a place of a piece, precise place in the code of T's, C's, and G's, which is code inside of each of the immune cells. This is a schematic of what it looks like with the structure of CRISPR, finding a place in the DNA and cutting it. And you'll see that there's this yellow strand here. And that's the sort of magic of CRISPR. CRISPR can be set to cut essentially any place in the DNA code by just changing out that yellow strand, which is RNA. And that we know basically exactly how to design those sequences, and they can now basically be ordered off the internet. And so if we can program this cut at a place in the genome, now all of a sudden, that might sound technical, but it's the basis for thinking about all sorts of changes. We can use those cuts to disrupt specific sequences, basically to get rid of genes that might be limiting the function of immune cell therapies. But we can also paste in new sequences. We can basically trick the cell into taking an extra piece of DNA and actually add a new piece of code. And we're using this now to put in codes for CAR T cells at precise places and other instructions that will make these cells more powerful and hopefully eventually more safe too. And the key to all this is that, we want to do this directly in a human blood sample. That's been the focus of our work, is that we take a human blood sample, use CRISPR efficiently, safely to make DNA instruction changes in exactly the cells that will go eventually into the types of cells that would go into patients to treat cancer and I think many other diseases. I'll just take 1 minute and show you what this actually looks like, because I do think that's important to give a sense of how easy this is. This is Ryan, who was a technician in my lab, basically straight out of college. He was taking blood samples, purifying them. And this is Jess, who was a graduate student. She could mix up a different CRISPR molecule. Basically, she would put a different RNA into each one of those little tubes and an extra piece of DNA if we want to paste an extra sequence into the DNA. And we could just basically pick and choose where we'd want to cut and paste by changing out those molecules in that liquid. So each one of those wells could get a different edit. And then the way we get these into the immune cells, we mix them with those T cells that were purified out of blood, and then we put them in this machine that gives them a little electrical shock. And this is a trick that's been used for a long time in biology. The truth is it's kind of black magic, but somehow that electrical shock will make the cells more porous. And we found that allows CRISPR to basically slide right in. And then CRISPR uses its own machinery to figure out where to cut in the DNA, and the cells start repairing it and the DNA cut and making changes. And so within a couple of days, we can actually see the changes. So some of the first changes, we would paste in sequences that would make the cells turn fluorescent green, and you could see that there. And we could just see how quickly and efficiently this process worked. But now that's not our ultimate goal. Our ultimate goal is to paste in sequences that give instructions to T cells to fight cancer. So we can put in instructions at a very specific place to make a CAR T cell, and then check to see that those CAR T cells that we've designed are actually able to recognize cancer, to discern cancer from healthy tissue, and to eliminate cancer cells, both in test tubes and in animal models of disease. Now, I'll just end by saying, it doesn't stop there. You see how easy CRISPR is. Now we've scaled it up even more. We're not testing things one at a time. We're testing every gene in the genome and seeing if we get rid of every gene or add every gene, which ones will boost the cancer-killing function of T cells. We do these experiments. There's roughly 20,000 genes in the genome. We test them all in a big pool and see which ones make better cancer-killing T cells. So we're really learning the rules of this DNA circuitry. So just to give you a sense of what this type of experiment looks like, now we don't make the changes one at a time, but each cell could get, for example, instructions for the same CAR T cell plus an extra gene, and we can read out which one of those extra genes will boost the cancer-killing ability of those T cells. And so we do this, and we can basically just watch and race those cells against each other. And now we start to see which ones not only find the cancer, but continue to overcome all of the ways that the cancer can fight back so that they continue to divide and be able to clear the cancer cells. So we are learning rules about how to make better CAR T cells incredibly fast. Plus, we have this ability to manufacture them using the power of CRISPR. Maybe I'll skip over this other than just give the example. This is one gene that we came out of these types of studies that we never would have found, but all of a sudden we can see much better CAR T cells. And normal CAR T cells here against a leukemia or against a solid tumor, at these doses, that you can see that the tumors shown in this glowing color persist, but when we get rid of this gene, RASA2, which had been previously unappreciated in T cells, we can make the T cells much better clearing these tumors, and the mice survive longer. And I'll just end by saying this approach is something that is bigger than my lab or any individual lab. This is an approach that many of us are now coming to realize. So we started a couple of years ago in the Bay Area, an institute dedicated to this. It brings together a nonprofit research institute, Gladstone, plus the large clinical campus and research institute of UCSF. And basically what we're trying to do is use the power of all the technologies that are available to us. We want to use the power of testing every gene in the genome. We want to use the power of the ability to read out the changes in genome with DNA sequencing, get lessons from human genetics, plus be able to use the power of computer science. Big data, AI, are allowing us to sift through all this faster and faster. And the goal is to really use these technologies to program cells to do what we want, so that they can recognize cancer cells, persist, divide more when they find the cancer cells, release things into the tumors that will allow them to go away, and then also be safe so that they'll turn off when the cancer is gone, maybe stick around as memory. So there's many things that we're trying to program into these therapeutic cells for cancer and many other human diseases. And we've assembled a team that's really thinking about how do we design all these programs and implement them. And the last slide I have here is basically just to say, this isn't just a lab project, this is advancing into clinical trials. We have a number of different facilities where we're actually doing these in ways that are approvable for actual clinical trials in humans. And that allows us to take the lessons from the lab and move them forward into clinical trials and hopefully develop cures faster. So I'm showing you here a pipeline for multiple different types of cancer that are being developed by not only our group, but others at UCSF and more broadly. And this happens academically. And I also mentioned that we're involved with Arsenal. So technologies from my lab and others have been licensed to Arsenal, and Arsenal has really assembled them into really powerful, promising treatments for a number of different cancers that are actually being manufactured in these facilities at UCSF, and patients are being treated around the country in 2 open clinical trials now. And with that, I just wanted to really show that this happens. These are my lab taking a break, but this is a much bigger pursuit of many, many labs around the world working together to try to take advantage of the opportunities. There's a lot of opportunity and a lot more to be done.
Amazing. Thank you so much, Alex, for sharing. At this point, we'll pass it over to Chris to give a little bit of background about his experience, as well as his experience with advocacy in this space.
All right, thank you so much, Hillary. Well, my name is Chris White. I mentioned I'm a mucosal melanoma survivor, thriver, and advocate. But in 2018, when I was 36 years old, I thought I had a hemorrhoid and didn't do anything about it. And shortly after that, I come to find a lump in my groin, and that's what got me concerned. I went to the doctor, got it checked out, and it turns out that that lump was a lymph node, and the pathology came back from that lymph node as metastatic malignant melanoma. I went to an oncologist who told me that this, my pathology was extremely rare, and asked if I wouldn't mind seeing an additional second opinion. I said absolutely, and so I live in North Dallas, and they referred me down to MD Anderson. I got down to MD Anderson, and at that point, I had already had, my initial tumor on my rectum removed that I thought was a hemorrhoid, and then also that lymph node. And they go in to tell me specifically that I had anal rectal mucosal melanoma, and that this type of melanoma is a rare subtype that makes up 1% of all melanoma. There's no known cause or standard of care, but there are a lot of advancements in treatments such as immunotherapy and such. And so, but we were going to be very aggressive with every treatment and, the way we approached it. And so from that point, I ended up going in and doing another lymph node dissection surgery, then followed that up by 5 sessions of targeted SRS radiation to my primary, to my rectum. And then I went to go do restaging for my scans. And I thought I was going to have a clear scan. And it turns out I went from stage 3C to 4. And so went immediately into combination immunotherapy, and I was scanning every 6 weeks. But the cancer was so aggressive, it went from my lungs, liver, and lymph nodes to now also in my kidneys, and was continuing to spread throughout my abdomen. So switched gears and went into chemotherapies plus single dose immunotherapy, continuing to scan every 6 weeks. It kind of seemed to put the Band-Aid on it for the moment. We went back to the combination immunotherapy, but at the same time doing radiation, because I had so many nodules that some of them were pretty big within my abdomen, and we wanted to attack those. So did 5 more sessions of targeted radiation, followed that up by 2 more sessions of combination immunotherapy of Opdivo plus Yervoy. And at that point, I then had about 6 combinations within less than 5 months, plus single dose immunotherapy. And I had colitis. And so that kind of put a brake on everything. Fought that for about 3.5 months, kept on relapsing with the colitis due to the prednisone taper program. And when I finally got recovered with that, I was too toxic for me to go back to immunotherapy, did some more chemos. And at that point, doctor was telling me that, we got to find an end game, because, we pretty much used every option that was on the board. And so that's when he introduced and asked me if we ever did a harvest or till. And I told him, I remember seeing the words on my chart, but no, we haven't. Please, describe it to me. And he goes to tell me that it's very similar to immunotherapy in that it uses your body's own immune system to attack it. But what they do is they take a tumor. And at this point, I actually had an additional lymph node that grew in my groin that would be able to be resected. But they take a tumor, extract it, send it off into a lab, and then they take those cells from the tumor, and then they grow those into the billions. And while you're waiting for those cells to grow, which is about a 22-day growth process plus a 7-day Q&A, you do a lot of tests from the patient, from an EKG, blood labs, baseline scans. I also had to do colonoscopy because of the prior colitis, make sure all the inflammation was gone. But there's a series of tests you have to go through, because about a week before they're till are ready to be infused to you, you have to enter the hospital and do a week of chemotherapy based upon 2 different chemotherapies to bring your white cell count down to 0, make your body's immune system a blank slate. Then they infuse the TIL via, it comes in an IV bag, just like, a bag of fluids. And then they follow that up with up to 6 doses of IL-2 or interleukin. And what that does is it basically like activates your cells to start running throughout your body like you're throwing a match on gasoline, and it starts to spread. And what I was sounded a little bit confusing, so I asked him to explain it to me again, maybe a little more simpler. And he said, look, you got to picture it like this, that, you need 1 tumor to extract and 1 to monitor. But they take those specific cells that mutated, and then they grow those into the billions. And you got to think of it like an army of, say, Pac-Man. And then when those are ready and they infuse it back into your body, those Pac-Man roam throughout your body and eat up all your cancer like the dots and cherries, no matter where the cancer's at. And so he was telling me that, this treatment has been around since the 80s. Dr. Rosenberg pioneered this out of the NIH and the NCI, and it's designed for solid tumors. And there's this is the treatment that I would need to do. Now, MD Anderson had done this for a very long time, but at the moment did not have a trial. And so he did say that there was a biopharmaceutical company that was in a late stage phase 2 cohort 4 trial, and that they were accepting patients, and I would qualify for it, and to track them down and get it moving. And so went to the computer and looked up Iovance Biopharmaceuticals, and there was about 57 worldwide locations, half of them here in the States. And I live in North Dallas, and I picked CU Anschutz in Aurora, Colorado. It's closest proximity to me, as well as I have some family still out here. And so came out to sign the consent in November of 2019. And then 2 weeks later, towards Thanksgiving of 2019, I did my colonoscopy, my tumor resection, and some labs. And then in the first week of December, they went in for my baseline scans. And a few days after that, I went in to go over the results, and my CTs were not looking great. But this time I finally had a brain metastasis, and that brain metastasis is an exclusion for the trial. And so the trial coordinator and my oncologist got together and discussed, what my options were, because, I was now excluded. But in order to get back into the trial, because it was closing, I needed to stabilize the brain metastasis. And the only way to do that, to be effective, to prove it was stable or in reduction, was to do 5 more sessions of targeted SRS to my occipital lobe. And I had a 1 centimeter Mets. And so they told me this on December 10th. But the trial, since it was closing, had a cutoff date, and it all backtracked from this date of January 15th. And that also had to include the washout period and everything. And so the very last date for me to possibly scan was on Christmas Eve of 2019. But before I could do any of the radiation, I had to get a mask made. In order to get the mask made, I needed insurance approval. And so it took up to 6 days to finally get approval. And then on the 7th, I got the mask made. And at that point, I didn't have 5 days to do 1 session a day. So I did all 5 sessions in 4 days. And then I scanned on Christmas Eve and sent the disc out to Colorado from Texas, and waited to go over the results, filled out my will, and the life. People were coming to visit me like that was the last time they're going to see me. It's very clinically declined at the moment. Came out to Colorado. They told me that, it was pseudo progression. It goes up before it comes down. It was actually 1.2, but they were able to prove that it was inflammation. So they let me back in the trial. So I had to wait, though, until the 8th to enter the hospital because of the washout period. And on the 8th, I entered the hospital, already had a port. They gave me a PICC line, started my week of chemo depletion, and got through all of that. And then on January 15th of 2020, they infused me with my TIL. And then I followed that up over the next 2 days and did all 6 doses of IL-2, and was finally released from the hospital on January 20th of 2020. So a total of 12 days in the hospital. And it turns out I was the very, very last person to be dosed out of 153 people in this entire trial. And so within the next 9 months or so, my labs and scans continued to improve. By my first set of scans at the end of February, I had 50-60% reduction throughout my body on all my tumors. And by September of 2020, they were telling me they thought I had a complete metabolic response. So I did a PET scan in October 2020, and then another one followed up in January 2021. And on that one, it was 100% clean and clear, zero uptake. And I've been scanning every 3 months ever since, and been clean and clear ever since. In fact, I'm actually in Colorado right now at CU Anschutz for my month 57. And my last follow-up for my 5-year follow-up comes this August. And so over the past few years, I have gotten greatly into being a full-time advocate and wanted to be an inspirational speaker. And so I've just been kind of self-taught. I self-published my book, Killing Cancer with TILs, tells my story over that was 18 months. But I work with patient advocates and philanthropy groups, anything related from melanoma to cell therapy, and then also with industry to speak into their internal teams that are in biopharmaceuticals with advanced cell therapies and specifically TIL. And so I stay real busy doing all of that. And I think the world of TIL, there's 7 approved cell and gene therapies, but only 1 for solid tumors. And but 90% of all cancer is solid tumors. And I believe, the most pre-treated patient that was in the trial for this. It was the C-144-01. And out of the 12 total mucosal melanoma patients, I was one of the 6 with a response. I'm very blessed. And that's about it.
Thank you so much, Chris. We really appreciate you telling your story. Alice, if you want to throw your camera back on, and we'll get into some of these questions. As a reminder, you can use the question and answer feature. We see questions coming in. Just a comment from somebody saying, cheering for your perseverance. Thank you for your dedication to research and advocacy. Another one that says, comment on Chris's amazing story and thank him for sharing it with such great detail. Really brings a lot of lots of research presentations and data to life. So glad he had such a great response. So wonderful to hear that. But we'll move into some questions. So maybe we'll start with some more scientific questions, thinking about this point. I think, Chris, you brought up the idea of cell therapies are largely for solid tumors. And Alex, do you have, from a scientific perspective, kind of the reason why have we largely seen them in, more in hematologic cancers? And is there a reason or a way that we're kind of thinking about bringing these amazing therapies to patients with solid tumors more?
I think there's at least 2 reasons. One is that as we design the targets for these kinds of engineered T cells, what they're going to go after, the target that's been designed on, or the now 2 targets that have been designed on blood cancers are really good targets. They capture they allow T cells to capture the cancer cells. There is collateral damage, actually. They get rid of a bunch of the healthy cells in the immune system, but those cells actually turn out not to be essential for life. And so people are able to live without those parts of the immune system. And so that has just been really well tolerated, and we don't have the same types of targets that we for solid tumors yet. Many people are working on it. The type of treatment that Chris got, the TILs, actually takes advantage of the fact that in some cases the body is able to find its own targets, and T cells work their way into tumors. And so that's actually a way of using the body's own immune system to teach us about the targets. And there's a lot of ongoing research about that. And it may take some combination of targets to put into these T cells. So that's one problem. The other problem is that even if we get targets for T cells to go into solid tumors have a bunch of tricks that evolve in the tumor to fight back against the T cells. And they can actually basically turn off and almost have like an immunosuppressed state locally inside of that tumor, so that the T cells, even if they find the cancer cells, might not be effective. And that's where a lot of the gene editing approaches that I talked about today could be really powerful. And we are advancing things into clinical trials now to basically fight back against the tricks of the tumor cell and to add resistance, add extra, boost the powers of T cells, even in those challenging solid tumor environments. So we need better targets and ways to boost the T cells. And luckily, there's tools and research that are racing ahead on both of those.
And maybe for both of you, and Chris, just from your advocacy work, maybe you know about this, and Alex, from, you mentioned some of the work that you're doing, but just from the field, what are some of the improvements that are happening beyond the approach that you're taking? Are there things that you know about in the field? And maybe we'll start with you, and then Chris, if you're aware of any, additional work that's going on too.
There's so much happening, and so we try to work collaboratively, and so I get a chance to see some brilliant research from other people too. There's a really exciting area of trying to design T cells in a number of different ways that can recognize more than one target. So one is that they could recognize tumors that have either this target or that target, or sometimes what we need is that in order to make targets available to keep them safe, we actually use combination of targets so that the T cells have to recognize two targets. And we think of that kind of like the two keys for a nuclear launch code, that you have to ensure that there's a double verification system before the T cells get engaged. And that allows us to pick targets that might not otherwise be safe, but we're really able to direct those T cells just against the cancer cells. So there's a number, there's really a suite of different approaches to get at those things. And it's moving fast. These are areas where new papers are coming out on this on a near weekly basis, and so there's a lot of momentum. People recognize the need.
So there's about 85 or so biopharmaceutical companies out there right now that are currently trying to get BLAs in front of the FDA, for solid tumors and for TIL. A lot of the companies right now, they're trying to figure out a way to activate the TIL after it's infused, other than using IL-2 and that sort of thing. There's a lot of work within the blood donation and tissue field, because in order to grow my specific TIL, into the billions, and there's no proteins added to it. So when they extract the tumor and send it off and grow it, it's the exact same cell that mutated. But in order to do that, they need feeder cells in order to pinpoint the exact one that goes down. And so, I know there's a lot of work right now in or autoimmune diseases like RA, diabetes, and that sort of thing. So, but also, places like the NIH and stuff, I know they're doing trials within, like, say, breast cancer. The sponsor that did my clinical trial, they're got current treatments right now for lung cancer and cervical cancer. And so, there's a pretty decent amount out there when you consider 90% of, treatment or cancers, is solid tumor. And so they're trying different ways, from everybody that I come across. I've actually talked to a handful of different biopharma companies that have reached out to me recently about doing some events with them, but, they have their own, version to where they can help improve like some of the, safety stuff to go along with it. But, when you do the actual treatment, all your side effects that go along, or at least for me, only happened while I was in the hospital. I've never had any long-term side effects from that treatment. All my long-term side effects were because of all the prior treatments I did before TIL. So, there's a lot of stuff going on there.
Yeah, and just thinking, you kind of mentioned the growth for your tumor cells and, getting your TILs grown up and all that kind of thing. Someone from the audience asked, is CAR T therapy always patient specific, or is there a possibility to create a generic, quote unquote, off the shelf version that could be used more universally?
Maybe I can answer that. The short answer is yes, there's a lot of efforts to make this off the shelf. I mean, I think it maybe the step back, there's even a bigger question of how do we make these treatments available? So, I mean, I think the hope, the power of this is that this is a living drug that we can put biological logic into, that can go into the body. It can take advantage of the fact that T cells are naturally evolved to protect us from infection. So we already are starting from a place of cells that have a sort of inherent evolutionary power. And then we try to make these cells stronger and be able to really go after the cancer cells without the side effects. That's the hope. And so we talk about all the research that's going into that. but to make a living drug requires a pretty complicated process. And I talked about it in a patient-specific way, but if that happens, the patient's cells have to go to a facility to grow them. The whole process can take about a month of going to the facility, testing them, making sure that they're safe to reinfuse, going back to the hospital, reinfusing them. And that takes time and money. And those can be limitations to patients getting these treatments. So there's a number of ways that people are trying to improve this. One is make the manufacturing easier. Maybe it's something that could be done in days or at the patient's bedside. So making it easier and more scalable is one. Two is people are trying off the shelf. Maybe it could be more like a medicine that you'd recognize, like a pill is not customized. It's something you go to a pharmacy and get. Could these cells be something that sit in shelves and are defrosted and infused when they're needed? And that would allow us to take all really good T cells that are uniform and infuse them in ways that might be more predictable. A bunch of technical challenges to get there, and there's pluses and minuses, and many people are working on trying to figure that out, but those are also advancing in clinical trials. And then the third, and maybe this is just an extreme of making this more available, is maybe these CAR T cells could be, or even finding ways to boost the actual TILs that are already in the tumor could be manipulated inside the body without ever taking them out. And so a lot of people are now working on thinking about technologies like lipid nanoparticles, which have become mainstream medicine since COVID, lipid nanoparticles or other types of nanoparticles, viral delivery that could be injected, could find their way to the T cells that they want to manipulate and give instructions to T cells inside of the body. And that would be another way to potentially massively scale access to these types of treatments.
Amazing. I think related to that, you kind of started to talk about access a little bit. And so, what can advocates do to help improve access to cell therapies? You just kind of walked through a lot of steps that are necessary from a scientific perspective, but are there other things to think about, as we talk to the advocates who are present today? Love to hear from you. And then Chris, I know you have plenty of experience with this, so would love to hear some of your experience and some of the things you're doing too.
Oh, well, maybe I'll let Chris go first on this. He's the expert.
Well, when you go to search the net, there's only so much out there. And so one of the things that I encourage anybody that has experience with this is, to share it, on multiple different social media platforms. Each audience will have their demographic, and so what I've come to discover is, the medical community, in a sense, they're very like Twitter or X. And if you're in the business, and or medical clinical community, you have LinkedIn. But a lot of the youth out there, they're very Instagram and TikTok. There's certain generations, they're only Facebookers, but, there's so many different areas. So, it's always about knowing. You don't know what you need to know until you need to know it. And if you don't know that clinical trials exist, or even there's such a thing as a cell therapy, then you don't even know to ask about it or inquire about it. And so, this being just now starting to get more movement within, the community on, autologous cell therapies. I'm also been speaking with some other doctors because, they're having insurance issues. It's not, well wide known that, you can do one treatment one time and have it work. Most everything, except if it's off the shelf or something, it's, you do it multiple times over, a period of time. And so, there's it, like I did my whole deal within the hospital setting, but just like a blood donor center where you go in and do like an apheresis, there a lot of work right now being done and being able to create these cell therapy centers to where you can do both your infusion, they can manufacture it, all within closer proximities. And when the FDA approved my specific treatment for TIL in February, there was only like 30 approved cell therapy centers. Well, now I believe it's a little bit over 40, but they should be up to 50 here pretty soon. But definitely trying to bring more facilities on. But you have to, all the doctors and everything, they have to have certain specific certifications in order to treat this, because it's very highly customized per patient. So there's all sorts of things with that as well.
Maybe I'll
Thanks.
just add from the perspective of a scientist, the role of patient advocacy is enormous. Chris and I met just a couple weeks ago, and I think it's so important for scientists who can get lost in their work at the bench to remember, to hear the success stories, to be inspired, and also to be reminded of the need. And so I think that this is a real alliance between scientists and patients to move this forward. And it's not just scientists, doctors, patients. It's also the regulatory agencies. We met in the context of talking to the FDA together to talk about how to design trials that serve patient needs, that are responsive to safety, but maybe also can go faster and take advantage of technological improvements. And so I think there's a real partnership that has to emerge between a number of parties to keep pushing as hard as we can to make sure that we're looking at the full picture of how do we use the available opportunities to, in the most responsible, but also the most effective way.
Absolutely. I wanted to add to that. For example, a lot of traditional ways of getting your information, whether it's, newspaper, TV, and or your internet, but being able to reach out and collaborate more with, different news channels and, try to find other, means of media to, just to get the conversations going, to where somebody asks, well, what's that? Like, I explain this, like, say, the average person goes in to see a movie and, there's a disclaimer either at the beginning or the end that said it's based on a true story. And, you didn't know that you were going to, learn something, until, that moment. But just by word of mouth, itself, can get so much more going. But just the patient community themselves, being able to show a lot of the clinical medical community, hey, we are proofs of concept. This does work. There's, there could be so much more done in this, but we need collaborative help. We need, there's doctors, medicines, facilities, like, all kinds of stuff. And so I just really try to encourage, anybody and everybody out there to, try to reach out, and be that proof of concept.
Awesome. Thank you. There's a comment in here, and I think it's related to what we've been discussing already, but just the idea of price. We know that these are really expensive. I think it ties into both of your points about, that bedside potential of, can we kind of do this in an easier way? Are there ways to use technology to kind of advance it? And I think Alex reminding us of, the regulatory piece is an important aspect of that comes into it. Is something that we kind of want to make sure that we're considering. It's all these different players. And so the more conversations we can have as a group, rather than, one-on-one or individually trying to find solutions, is going to be important. Another question about kind of the oversight for ethical research development. What are some of the things that are considered? We've talked a lot about positives, but we know that there's a potential downside or risks that are involved in some of these therapies. And so how are we thinking about, ethical research using CRISPR in industry and at the bench too?
Yeah, I mean, I can kick this off, but I'm very curious to hear Chris's perspective. I think ethics comes into everything, and ethics is pretty far reaching here. I think access is one of the major sources of ethical challenge, right? I think we see this, for example, it's really pronounced right now, where not in cancer, but in sickle cell disease, where there's an extreme of this, where all of a sudden CRISPR has basically cured sickle cell disease. I mean, there's now the potential to cure sickle cell disease. It is extremely expensive, and only a small number of patients will be treated, and probably, really not in the centers where a lot of the patients have the greatest need, especially in Africa. So I think as technologies advance, thinking about access is something that we have to weave into conversations early. And that's a mix of payment plans, but also technological plans of how do we think about scaling things up. So I think access is a major source of ethical concern. And then another one is, maybe I'll stop there. And there's no shortage of I think thinking about how are we designing treatments for patients with the greatest need and not being distracted by a lot of things. making sure that researchers stay focused on that, and share data. Oh, I know the other thing I was going to say. The other one is, I think this is true for any new area of science, is thinking about how to bring patients into the conversation about honest and informed consent. That everything, everything I talked about, everything we're working on, there are massive unknowns. And there are risks. If we boost the immune system, there could be side effects of the immune system going out of control. CRISPR could cut and have unexpected effects. So we try to balance and think about where there are opportunities, where the benefits really are likely to outstrip the risks. But patients need to be involved in that and doing that ethically and responsibly, and not letting financial interest of people developing these treatments crowd that out, are all things that we have to be vigilant about.
I often tell people, would you want to fight multiple battles, or do you want to win the war? Do you want to slowly rip your Band-Aid off, or do you just want to just get it off? From a patient, myself and many people that I've known, they're willing and wanting to do anything and everything they can to live. And so they're really just looking for a chance. If they're paying all their premiums and out of pockets and doing everything on their part, the cost to do a one-time treatment for, like what I did, could be upwards to, maybe $500,000 or so. But if you do another treatment, that's over 18 months or so, that could almost add up to the same. And so, but if a patient waits until, they're already heavily pre-treated and then they get into it, they've taken, massive damage, and hits, to where by the time they do get to the process to be able to do a cell therapy, they might physically not be able to do it. And, or, some of them have been mentally beat down, right now till, is a second approved, treatment after, you fail on one. But the sooner you can get to it, it would, I believe, would be, the more beneficial, because, the body's like healthier to, be able to attack like any possible side effects. Now, when you sign a consent, just like anything else, it'll always have a list of possible side effects, but sometimes, they'll say they might happen, they just don't know until you're there. But, and when you do, something like till you're in the hospital for a couple of weeks, they have all the, infrastructure set up to where if they need to, they can help you. Like I've known a lot of people have had to go to ICU. I myself didn't have to, but, I believe I had to get one transfusion for platelets. That happens quite a bit, but they're set up for it, fluids, pain meds, that sort of thing. And so, it's always going to be risky, but it's even, it's one of those things where you're only going to miss 100% of the shots you don't take. And so it is kind of a, not, I won't say a fine line, but there is it's an odd situation, to try to weigh out, like, is this safe, and that's why it's a customized per individual, how healthy are they going into it? How aggressive has their disease burden already, become on them?
Thanks, Chris. Yeah, I think the idea of a kind of a individualized, we talk a lot about precision medicine. It's kind of the most precise precision medicine. And so having not only the treatment itself, but some of those additional aspects of, platelet infusions and those kinds of things afterwards for continued oversight. You are how far out? It's almost 4 years. How long do you have to be on the trial is where I'm going with
so
this.
I was dosed January 15th of 2020, and I've been doing my scans every 3 months. And so it's a 5-year, consent, or up to 60 months, but those months are based on 28-day months. And so, after this call, I go in and do my brain MRI and, full body CT and head neck CT. But this is for month 57. And then in the middle of August, I'll go back for month 60 and my initial 5-year consent is over.
Wow.
And then we'll figure it out from there. I mean, to me, it's almost scarier, because right now I'm going 4 times a year, give or take. But to go from 4 times a year to say, like 2. So I'm trying to, we haven't figured out what the long-term thing is going to be, but told them I'd be a lot more comfortable with going from 4 to 3, at least for a year or so, and see where it goes. But I think I've had over about 100 some odd scans in the last like 5 years, if you add up PET scans, CTs, MRIs, so things like that.
Yeah, and I think kind of we're kind of coming close to the end, but one of the questions we received was about the expanding, we kind of touched on this, but expanding to the developing world, but also expanding to smaller rural hospitals. Travel can be expensive and prohibitive. And I know, as you mentioned, you're currently in Colorado, live in Dallas. So you're flying out once a quarter to get your scans because the clinical trial wasn't offered in a broader capacity. So is that that's another thing potentially that advocates can kind of have conversations about, to Alex's point, being involved in the clinical trial design, but also site selection, ensuring that there's a broader kind of access for that too.
And well, one of the things I try to advocate for is, prior to the trial, I lived in Dallas, but I was also going down to MD Anderson. And so, I call it like I had, multiple captains, but only one captain of the ship. And so, MD Anderson was making the calls. And so when it came to surgeries and radiations, I definitely was doing them there, but he would prescribe, the systemic treatments, whether it be any kind of immunotherapy or chemos. And I got my oncologist, in Dallas, be able, to get them to communicate, work together, even though they're from different organizations, because, I'm the patient, I'm the one paying everybody, and it was like, I had to put together, my team. But when you're doing a systemic treatment, I mean, that can be done anywhere that has an infusion center. And so, it wasn't necessary for me to do that, but that actually saved me a lot of trips going back and forth to Houston. I was already doing that quite a bit. Now, since I did my TIL in January of 2020, shortly right after that, COVID hit. And, the way the consent was designed for this specific trial, and, I agreed to it and everything, was, I needed to come back here on site to do my scans and follow-ups. And one of the reasons that, let alone from it being closest to proximity, being a 12-hour drive or like an hour and a half flight, was I have family here. So I had some sort of setup in regards to, I have somewhere to stay and they could help feed me and so forth. I just needed to get here. But when COVID hit in March of 2020, I was allowed to scan in Dallas, FedEx the disc, and then get on a Zoom call to go over the results. And that worked great until the restrictions let up. And then I had to come back on site. And so I'm so committed to, the trial and my data that, it's one of those, it's become part of my life to travel, to come do this. But, they're for future, trials and things, collaborative partnerships between oncologists, because at the end of the day, they're all working to save the life of the patient. So, but if you have different types of organizations and, states and insurance, there's so many different things that go into it. Those conversations need to be had so that for, things like telehealth and, once you get the initial treatment, your follow-ups, can be done virtually.
Hearing Chris talk was a reminder to me of just how difficult the system is to navigate. And I hear this all the time of like, from center to center, you might not hear about some trials. You really it's amazing how much work patients still have to do to go out there and really find what's available and what's experimental, what's promising. And so, I think we have to do a better job of helping patients navigate the system to make sure that the outcomes are more equitable, regardless of where you are.
Yeah, make the system better. That'd be great. And then as we wait for that to happen, let's make sure that we're providing those resources. I think, related, there's some questions about finding trials. Are there any suggestions or ideas for how people could go and find clinical trials that are related to cell therapies?
So you can go to clinicaltrials.gov. You can go to, I think it's CRISPR, like that organization, CRISPR. If you do just a Google search, you'll find a handful of different organizations. Like, for example, if you were to do, TIL or Amtagvi, you can go straight to Iovance's or Amtagvi's website, and it'll, they plug in your zip code and it'll tell you the closest proximity of an approved center. But if you just go to clinicaltrials.gov and you just start entering cell therapy or TIL, and just start playing around with the different types of therapies, I mean, you'll be surprised both, nationally and internationally. Currently, I know that Iovance is, they're working on Tilvance 301, which is, another global treatment or clinical trial, though, to where they're doing TIL, but they're also doing like Pembro, Keytruda. So if you do the arm where you do Keytruda and it doesn't work, you still have the option to do TIL. But it's just what I was saying earlier, you don't know what you need to know until you need to know it. So one of my kind of goals is to like indirectly be like a influencer or whatever, like if I can create so much content that when somebody goes to Google anything about rare disease, rare cancer, clinical trials, they're, something positive will pop up, because I didn't have that when I was going through it. And if enough patients out there, use their voice to do so, then it'll organically grow. So
Thanks, Chris. Yeah, very helpful.
I'm not an expert on navigating this exactly, although I often end up talking to people about it. Parker Institute for Cancer Immunotherapy, which I'm involved with, PICE, has also been very generous and helpful in helping patients navigate this and connect research, and also point patients towards the right experimental centers.
Thanks, Alex. Great call out. Well, we're at the end of our webinar. We'd like to say a major thank you to Alex and Chris for joining us, not only on May 6th, but also today to have this additional discussion. We are open to feedback. This is our first time doing a webinar related to this topic, kind of like this, and our hope is to continue conversations like this. So please feel free to email us, advocacy@focr.org, if you have any additional thoughts. We'll be sharing a survey following the event. But as always, we appreciate your time and look forward to continued discussions. Thank you so much for joining today.
Thank you.